my name is Katerina Jordan, and I'm a researcher in the Department of Plant Agriculture at the University of Guelph. My areas of research focus include plant pathology and sustainable turfgrass management. Today we will be discussing a project that allows me to combine both areas of research as we delve into the sustainable and alternative methods for management of take-all patch. Take-all patch has historically been thought of as a disease of new or recently renovated turf that tends to go away after 5 to 7 years. This is thought to occur because of a buildup of natural antagonistic microorganisms in the soil that are able to outcompete the take-all pathogen, leading to an eventual decline in the occurrence of the disease. However, in our lab, we've noticed over the years that we were seeing take-all on golf course greens that were 10, 15, and even 20 years of age. We started gathering data on occurrence throughout Canada and noted that the disease seemed to appear on older greens primarily in samples from Ontario and west through to Alberta. This led us to believe that decline of the pathogen may not be occurring on alkaline soils or soils where the pH is above 7. In addition, we noted that some samples that had the classic symptoms of take-all patch were not being identified by the university's pest diagnostic clinic through DNA analysis as Gamenomyces aveni, the commonly known pathogen of the disease. We then began to wonder if perhaps we had more than one causal agent of the disease here in Canada. Thanks to funding from the Ontario Turfgrass Research Foundation, we've initiated a study to answer some of the questions that these observations brought up. Here to discuss some of the details of the project is graduate student Karen Francisco.
Hi, I've been working with take-all since my undergraduate degree, and I'm continuing it with my master's, and I'm hoping to continue working with take-all for my PhD.
Slopiomyces cylindrosporus was identified as a possible causal agent of take-all patch. After inoculating healthy bentgrass with Slopiomyces cylindrosporus, symptoms of take-all patch appeared and runner hyphae were observed on the roots. The next steps of this project is to isolate the fungus from the roots and perform DNA extractions on the pure cultures. After that, the DNA will be sequenced. Currently, no research has been published describing an effective method to inoculate a field with Slopiomyces cylindrosporus or Gamenomyces aveni. In 2 greenhouse experiments and 1 field experiment, we were testing 3 methods of inoculation. The first method of inoculation is to apply the inoculum to the surface of the turf. The second was to remove a plug and fill the hole with inoculum. The third is to add a layer of inoculum 5 centimeters below the surface of the soil. Once symptoms of take-all patch appeared, the roots were assessed for the incidence or the presence of runner hyphae and the severity of the runner hyphae. The field trial is still running, but for the 2 greenhouse trials, the incidence of runner hyphae and the severity of runner hyphae for the plug and layering treatments was significantly larger than the surface treatment, resulting in higher incidence and severity.